Bismillahir Rahmanir Rahim. Welcome again to our next video about why it's dangerous to have our DNA or our chromatin extracellular. Our video today will be about the role of extracellular chromatin in diseases. And again, my reference today is this nice paper that was published in Cell Death and Differentiation in 2023, which its title is Moonlighting Chromatin When DNA Escapes Nuclear Control. So we will first talk about immunothrombosis and the role of extracellular chromatin in the process of immunothrombosis. Actually, over the past 10 years, it has become more clear that neutrophils, particularly NETs, play a significant role in the development and the progression of blood clot formation in various disease scenarios. And that's why the term immunothrombosis has been introduced to emphasize that the connection between the body's innate immune system and the abnormal blood clotting. The activation of thrombosis by components of the innate immune system provides an evolutionary benefit by physically trapping infectious agents within blocked blood vessels and restricting their spread through the circulation. However, excessive immunothrombosis plays an important role in worsening of various pathological conditions such as sepsis-related coagulopathy, necroinflammation, and severe cases of COVID-19. Also, in other diseases like pancreatitis, it has been demonstrated that neutrophil extracellular traps or nets can obstruct not only blood vessels but also glandular ducts. In addition to their role in physically entrapping microbes, immunothrombosis also serves advantageous hemostatic functions during episodes of mucosal damage in acute ulcerative colitis flares. In this scenario, the absence of PAD4, which is an important factor of netosis process, is linked to mucosal blood loss. So in addition that we have a bad scenario in some diseases, this immunothrombosis in others can serve on the beneficial side. There are numerous interactions between neutrophils, components of neutrophil extracellular traps, and key elements involved in thrombosis, such as platelets, serine proteases in coagulation cascade, fibrinolysis, and fibrin mesh itself. Neutrophils and NET engage with these elements through various mechanisms, including receptors located on the cell membrane, effector proteins released from granules, chromatin found in NETs, including nuclear and granular proteins bound to NETs, as well as extracellular vesicles. So they make connection with blood coagulation factors through different ways. Actually, platelet activation can stimulate the generation of neutrophil extracellular traps or nets. On the other side, platelets also have the ability to attach to and aggregate on the extracellular chromatin of nets. These aggregated nets, known as egg nets, serve as a framework for the formation of blood clots and can cause blockage in both blood vessels and ducts. Similar to the von Willebrand factor, nets provide a surface for platelet adhesion and the initiation of coagulation. The blockage of small blood vessels in organs along with net-induced endothelial dysfunction can lead to impaired blood flow in capillaries, potentially resulting in organ failure and increased risk of mortality. In this diagram, we can understand the process of immunothrombosis where neither thrombosis nor inflammation occurs alone, as I already described in another video. We can see here how the nets share with the von Willebrand factor to make a platform in which platelets and other factors will attach. We can also see how the innate immune cells like monocytes and neutrophils share in activation of thrombosis, and on the other side, platelets activate the immune cells. As a result, aggregated neutrophil extracellular traps or agnets play a role in the development of multiple diseases. And as previously mentioned, nets can obstruct blood vessels in individuals with COVID-19. Also, occlusions associated with nets have been observed in coronary vessels during myocardial infarction and atherosclerosis, as well as in cerebral vessels during ischemic stroke. Apart from cellular interactions that can promote the formation of neutrophil extracellular traps or nets and pathological thrombosis, researchers also are investigating the role of soluble mediators that share in this process. Particularly, they give attention to what's called PAD4, as it is believed to connect inflammation and thrombosis. And actually, studies have shown that the injection of recombinant human PAD4 triggers the formation of von Willebrand factor platelet strength in mesenteric venules. PAD4, which is the abbreviation of protein arginine deaminase 4, plays an important role in mitosis by catalyzing citrullonation of histones, and also PAD4 activate neutrophil inflammasomes. Another important function for PAD4 is the mediation of citrullonation of what's called ADAM-TS13. This is an enzyme responsible for cleavage of von Willebrand strength. So PAD4 inhibits this enzyme, resulting in permitting platelet strength to persist near the vessel wall. Also, PAD4 guided thrombin activation may then facilitate thrombus maturation. And also, citrullonation has a significant post-translational modification that affect proteolysis process. So in this diagram, we can see how it is important, this PAD4 in the process of immunothrombosis. PAD4 share in the process of mitosis by citrullonation of histones, and PAD4 also stimulate inflammasomes and the release of inflammatory factors from neutrophils. PAD4 also inhibit what's called ADAM-TS13, the enzyme responsible for cleavage of von Willebrand factor, and PAD4 guided activation of thrombin will lead to thrombus maturation. These findings offer potential explanations for the ineffectiveness of the traditional anticoagulants in preventing blood clot formation during specific septic conditions and in disseminated intravascular coagulopathy. Pan-PAD inhibitors and PAD4 specific inhibitors are already important in investigating NETs and immunothrombosis and will be further explored as a potential therapeutic choices in a broader sense. Now I'm going to talk about the role of extracellular chromatin in lung diseases. So in chronic lung diseases like asthma, which was traditionally being categorized as an eosinophilic disease, recent findings have referred to the involvement of neutrophils in this condition. Similarly, patients with chronic obstructive lung disease, pulmonary disease, COPD, show significant airway inflammation dominant by neutrophils. Elevated blood neutrophil counts also have been linked to higher mortality rates in COPD patients, and in both lung diseases, asthma and COPD, the presence of neutrophil extracellular traps has been observed in the airways, indicating their association with the inflammation. And in the case of COPD, NETs also are associated with airflow limitation. In cases of acute lung injury and acute respiratory distress syndrome, higher levels of neutrophil extracellular traps in the bloodstream have been linked to increased severity and mortality. Also, lower levels of DNase1, DNase1 is the enzyme responsible for degradation of the NETs in the plasma, have been associated with the development of sepsis-induced acute respiratory distress syndrome. These findings highlight the importance of maintaining a delicate equilibrium between net formation and degradation to prevent lung injury. Cystic fibrosis is a condition caused by genetic mutations that result in impaired hydration and clearance of mucus, leading to persistent pulmonary infection and inflammation, primarily done by neutrophils. The sputum of cystic fibrosis patients contains a sizable amount of neutrophil extracellular traps nets and proteins associated with nets. The activity of neutrophil elastase and the presence of myeloperoxidase are correlated with the progression, severity, and improvement in lung function observed in cystic fibrosis. However, we have to remember that the bad outcomes of disease are not only related to the formation of neutrophil extracellular traps, but actually to an imbalance between net formation and degradation. In a murine model, it was demonstrated that complete deficiency of PAT4, the enzyme involved in net formation as I described before, led to a reduction in net formation and subsequent lung injury. Because this reduction was accompanied by an increased bacterial load and inflammation, highlighting the important role of neutrophils in infections. Neutrophils actually also play a role in orchestrating later stages of repair. They promote epithelial cell proliferation and release proteases necessary for the processing of collagen essential for scar formation. Now I'm going to talk about the role of extracellular chromatin in autoimmune diseases. So while neutrophil extracellular traps play an important role in eliminating pathogens, they are also playing a role in the development and pathogenesis of various autoimmune diseases. For example, rheumatoid arthritis, systemic lupus erythematosus, anti-neutrophil cytoplasmic antibody-associated vasculitis, antiphospholipid syndrome, psoriasis, and others. Actually, imbalance favoring net formation over degradation by DNase can lead to the accumulation of released chromatin and associated proteins in the extracellular matrix and interstitial spaces and lumens of blood vessels and ducts. This accumulation contributes to the progression and manifestations of these autoimmune conditions. In these scenarios, the released nuclear components can act as origins of autoantigens, potentially triggering the production of autoantibodies and immune complexes. This process becomes more significant when the released material undergoes post-translational modification such as oxidation, citrullonation, carbamylation, or the creation of new epitopes through proteolytic cleavage. These modifications enhance the immunogenicity of the nuclear constituents, contributing to the development and progression of autoimmune responses. And the autoantibodies generated in these autoimmune diseases target a wide range of disease-specific antigens which present significant variability. Examples include double-strand DNA in systemic lupus erythematosus, citrullinated proteins in rheumatoid arthritis, anti-lysosome associated membrane protein 2 and anti-myeloperoxidase in anti-neutrophil cytoplasmic antibody-associated vasculitis, and phospholipids in antiphospholipid syndrome. These diverse antigens represent distinct targets for the autoimmune response in each respective disease. In addition, these autoantibodies have the ability to influence the persistence and immunogenicity of neutrophil extracellular traps themselves. So by binding to chromatin structures, these autoantibodies stabilize nets and prevent their degradation by DNases. Also, in patients with systemic lupus erythematosus, the complexes formed between anti-double-strand DNA antibodies and nets stimulate the secretion of type 1 interferons by mononuclear phagocytes and activate NF-kappa B in endothelial cells with involvement of the Fc gamma receptors. So these interactions will enhance inflammatory immune responses and share in the progression of disease and autoimmunity. The formation of neutrophil extracellular traps within the capillaries and the formation of aggregated nets in larger vessels have activating effects on endothelial cells and the coagulation cascade, leading to platelet aggregation. Uh, as I described before in immunothrombosis, in conjunction with autoantibodies, nets contribute to the formation of immune complexes and promote thrombus formation. This process can result in the occlusion of blood vessels, particularly in the microvascular bed, leading to organ damage and, in severe cases, fatal outcomes. Also, nets modulate immune responses by interacting with other immune cells and humoral components. So, like histones and other net-associated proteins act as damage-associated molecular patterns, and so contributing to the immune activation observed in these conditions. Also, nets activate inflammasomes and the complement system through classical, alternative, and lectin pathways, as well as the coagulation cascade. Also, the formation of neutrophil extracellular traps by neutrophils in the spleen triggers immunoglobulin class switching, thereby affecting B cell responses and shaping the immune system towards antibody production. In systemic lupus erythematosus, also the formation of LL37 DNA complexes, as I explained before in another video, triggers the activation of self-reactive memory B cells, leading to the production of autoantibodies. Neutrophil extracellular traps also, or nets, play a role in lowering the activation threshold of T cells and also induce the production of type 1 interferons by plasmacytoid dendritic cells, which are characteristic cytokines, these interferons, of systemic LE. This creates a self-reinforcing cycle where dysregulated net formation and inflammation are interconnected, as certain responses mediated by nets promote further recruitment of neutrophils and the formation of additional nets. In the scenario of anti-neutrophil cytoplasmic antibody-associated vasculitis with microscopic polyangiitis and systemic lupus erythematosus, the self-reinforcing loop that I mentioned before is intensified due to diminished DNase 1 activities. This leads to accumulation and aggregation of remnants derived from neutrophil extracellular traps. The reduction in DNase activities can occur due to genetic deficiencies or consumption of the enzyme or the presence of circulating inhibitors or autoantibodies that impair the function of DNase, as observed in patients with sporadic systemic lupus erythematosus. Consequently, strategies aimed at prevention of net formation through the use of inhibitors or promoting net degradation by introducing DNases represent a potential therapeutic approaches for the management of chronic autoimmune diseases driven by nets. Now let's talk about the role of extracellular chromatin in exocrine ducts and stone formation. Actually, the innate defensive role of neutrophil extracellular traps have a role in the progression of obstructive and inflammatory diseases that affect the ducts of exocrine glands. Neutrophils play a physiological role in watching out the ducts of exocrine organs, and various factors can trigger the formation of nets and the aggregated nets within these ducts, including alterations in ion concentrations or pH levels, crystal precipitations, the presence of bacteria, or the presence of foreign bodies. These diverse triggers can initiate the process of net and egg net formation within the ductal system and potentially can lead to the development of obstructive conditions and subsequent inflammation. Regardless of the underlying cause, the formation of aggregated neutrophil extracellular traps or agnets leads to a decrease in the excretory flow and subsequent accumulation of occlusive substances within the nets. Over time, this results in gradual development of secretory stasis, creating an environment favorable for the formation of calculi. This process sets a vicious cycle characterized by further obstruction of the ducts, reduction in flow rate, and inflammation of the surrounding gland or tissue. Examples of organs and tissues where this phenomenon has been reported include pancreas, gallbladder, tooth supporting tissues, ocular glands, and salivary glands. Aggregated neutrophil extracellular traps or agnets acts as adhesive agents, incorporating crystals, pathogens, cellular debris, and viable immune cells. They promote the consolidation of these materials, facilitating the formation of tophi and calculi observed in conditions such as gouty arthritis, cholelithiasis, sialolithiasis, and meibomian gland disorders. As regard gouty arthritis, in gouty arthritis, there is a precipitation of uric acid as monosodium urate crystals in joints, triggering acute inflammation. Macrophages engulf these crystals, activating the inflammasome and releasing pro-inflammatory cytokines, leading to extensive neutrophil recruitment. Neutrophils binds to the crystals, inducing net formation and releasing pro-inflammatory mediators like tumor necrosis factor alpha and interleukin 6, which contribute to oxidative stress-induced inflammation. Elevated neutrophil counts prevent the proper degradation of nets by DNases. Consequently, the nets co-aggregate with crystals, leading to the formation of large tophi. Once the initial pro-inflammatory response subsides, proteases within the tophi aid in resolving inflammation by breaking down specific cytokines and chemokines. As regard the role of extracellular chromatin in periodontal crevice and periodontitis, neutrophils initially encounter in dental biofilms within the gingival crevice, which is the space between the gingival and tooth surfaces. This crevice is filled with periodontal crevicular fluid. It translates from blood plasma. In periodontitis, there is excessive formation of neutrophil extracellular traps in this fluid. Also, the crevice contains numerous outer membrane vesicles from the dental biofilm, which are internalized by neutrophils in the crevice. Actually, outer membrane vesicles play a critical role in bacterial colonization, virulence, factor delivery, and pathogenesis. Bacteria produce these vesicles to manipulate the host's immune response, enabling them to evade host immunity. Pre-activated neutrophils that infiltrate the gingival epithelium encounter and internalize numerous outer membrane vesicles loaded with lipopolysaccharides when they enter the periodontal crevice. These extracellular vesicles, which contain bacterial outer membrane components, activate the caspase 411 and gas-dermin D signaling pathway in neutrophils, leading to the formation of nets. Also, neutrophils, it was found that neutrophils deficient in caspase 4 and 11 produce fewer nets compared to the wild type control. And also they found that toll-like receptors 2 and 3, 4, 7, and 9 are not necessary for net formation by neutrophils stimulated with the outer membrane vesicles from oral pathogens in vitro. As regard the role of extracellular chromatin in ischemic diseases, actually sufficient oxygen supply and pH changes in the microenvironment impact net formation. Tissue alkalosis promotes net release, while acidic conditions decrease net formation. And tissue hypoxia often accompanied by metabolic acidification, so there is a decrease in net formation. And tissue hypoxia is regulated by what's called hypoxia-inducible factor 1 alpha in human tissues. Hypoxia stabilizes and translocates hypoxia-inducible factor 1 alpha to the nucleus, inducing the transcription of hypoxia-related genes. In neutrophils, nuclear hypoxia-inducible factor 1 alpha upregulates the NF-kappa B transcription, prolongs the viability, and enhances the degranulation and chemotaxis of neutrophils during hypoxia. So, in acute myocardial infarction, neutrophils with a high nuclear hypoxia-inducible factor 1 alpha content remain viable, while those with low levels form nets, and by staying viable in hypoxia tissue, neutrophils can phagocytose and clear cell debris, contributing to wound healing. As regards the role of extracellular chromatin in cancer, elevated net levels in tumors are associated with poor prognosis in cancer patients. They correlate with higher tumor grade, disease progression, metastasis, and decreased survival rates. The presence of NET is often concluded from the elevated levels of myeloperoxidase DNA complexes in the serum and to a lesser extent detected directly in tumor tissues. NETs can contribute to the formation of metastases by capturing tumor cells at distant sites through the binding of coil-to-coil domain containing protein 25, which is expressed on specific cancer cells such as colorectal, breast, prostate, and liver cancer cells. Also, NETs play a role in regulating various important processes associated with cancer development and metastasis. These processes include creating an immune-evading microenvironment, activating dormant tumor cells, facilitating tumor cells extravasation, promoting angiogenesis, and increasing vascular permeability. Now I'm going to talk about the role of extracellular chromatin in ocular diseases. NETs play an important role in antimicrobial responses in the eye. However, they are also claimed in eye pathologies. Severe cases of chronic dry eye disease in graft versus host disease have shown an association with NETs. Also, NET formation is believed to be induced on the ocular surface of dry eye disease patients in response to hyperosmolar stress. NETs contribute to molecular pathological changes in patients with corneal injuries. The choroidal and retinal compartments are also susceptible to the effects of NETs. In Behçet's disease, a form of non-infectious uveitis, there is increased NET formation that has been observed, potentially contributing to the extended vasculitis associated with this condition. Also, both in vivo and in vitro data suggest that elevated blood glucose levels in diabetic retinopathy, a leading cause of irreversible blindness globally, can induce the formation of NETs. Now I'm going to talk about the relation between extracellular chromatin and adhesions. Peritoneal adhesions frequently occur following abdominal surgeries and are associated with significant complications, including intestinal obstruction, pelvic pain, and infertility, impacting the quality of life for numerous patients worldwide. Recent findings indicate that the formation and aggregation of NETs can exacerbate wound healing, prolonging the inflammatory phase, and activated neutrophils have been observed in burn patients long after the initial thermal injury, and DNases have shown potential in accelerating dermal wound healing in both mice and diabetic patients. So, opposite to the traditional belief that inflammation is crucial for wound healing, oral wounds with minimal neutrophils, macrophages, and T cells healed faster and had minimal scarring. The underlying mechanisms of adhesion formation, despite its clinical significance, remain poorly understood. Adhesion formation involves processes such as hemostasis, angiogenesis, and tissue remodeling. Inflammation is orchestrated by the innate immune system and appears to play a key role, with neutrophils being rapidly recruited to sites of bacterial infection or tissue injury, engaging in phagocytosis, degranulation, and NET formation. A recent preprint indicates that abundant NET deposits associated with fibrin contribute to the formation of adhesions in both mice and humans. The use of DNases to digest extracellular DNA prevented the development of adhesions following surgical procedures. These findings suggest that nets serve as the initial platform and the attachment of fibrin reinforces the structure, leading to formation of mechanically strong adhesions. Mice lacking BAT4 or treated with DNases show significantly reduced adhesion formation. As regard the role of extracellular chromatin in spinal trauma, actually the pathophysiology of traumatic spinal cord injury involves two distinct phases. The primary phase, which involves the mechanical injury that damage axons, neuronal cells, glia cells, and the blood-brain barrier. Following this initial trauma, neutrophils accumulate locally within minutes and initiate a secondary phase of damage by releasing nets. This exacerbates cell damage and worsens the severity of neurological deficits. Secondary injuries associated with traumatic spinal cord injury include vascular damage, disturbed hemostasis, edema formation, and inflammation. Myeloperoxidase, a characteristic enzyme of NETs, enhances the levels of pro-inflammatory cytokines such as interleukin-6, interleukin-1 beta, and tumor necrosis factor alpha. In a mouse model of spinal cord injury, the absence of myeloperoxidase reduced neutrophil infiltration and expression of pro-inflammatory cytokines and apoptosis. This led to improved motor recovery in myeloperoxidase knockout mice. The findings suggest that myeloperoxidase contributes to secondary injury and exacerbates tissue damage after traumatic spinal cord injury. So anti-NET therapies targeting PAT4 or DNase1 show promise in reducing NET-induced neuroinflammation, traumatic spinal cord injury-associated edema, glial scarring, and fibrotic scarring. Topical applications of DNases during surgical decompression of the spine holds great potential in reducing secondary injury and scarring. And pharmacological modulation of neutrophils during the extended surgical window or the use of heparin known to take apart nets and limit net formation are additional treatment options that worth exploring. I hope that you have enjoyed this video and you can now have a deeper understanding of the process or the effects and dangers of extracellular chromatin in different diseases. And I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.